Okay, take it away.
All right, I'll just start. Morning, everyone. I'm Krichmar. I guess I'm kicking off the meeting with this first speaker. This is collaborative work with Doug Nitz's group at UCSD here and Liz Crastel at UCI. I'm at UCI as well, UC Irvine. What we're working on is this perspectives between global maps. I'll explain what that means, and a first-person perspective. And we're looking at it in multiple ways. But just to give you an idea, it's not too bad. We do this all the time. So you look at the picture on the left, there's a person looking at a map and the woman pointing. So the person is doing a global map perspective of where they are. And the woman is doing a first-person perspective, like, oh, on this map, I should be seeing this building. If I'm looking this way, or if I'm looking this way, I should be on this position in the map. And I would say we do that in our head, and so do animals. So bats are incredible navigators, and they're up in trees, especially fruit bats, and looking down and flying all around. You've ever had a bird feeder with a squirrel on trying to get into it? You know, they must be doing some sort of three-dimensional perspective transformations. So we wanted to get at this. We did some original work with modeling. We used a variation autoencoder. And what we would do is the global map in this robot simulation that's running, the little green dot that's moving is the robot. The inset, which you can see is the camera on the robot. So that's the first-person perspective. And then the global map perspective is the overhead projector in this simulation. And so the variation autoencoder was to take the scene from the camera and reconstruct the scene that would be from overhead and vice versa. And just by doing that and examining the latent variables in the autoencoder, we found place cells and head direction cells. So if you're going from the first person to the global direction, we saw really good head direction cells and things that a neurophysiologist would call a place cell, but not really sharp. If you went the other direction, you saw head direction cells that weren't as good, but you saw better place cells. So this was a really good start that actually kicked off talking with Doug and Liz's group that, hey, we should try this experimentally because there wasn't much data on it. And that study had some limitations, though. So it was trained on static pairs of images. And we all are constantly moving, seeing the world as a stream of information. And there's certainly in the hippocampus and other areas, the medial temporal lobe and spatial areas, this sequential temporal processing. And we only look for place cells and head direction cells. And there might have been other spatial neural correlates. And there was no way in this model to actually examine what visual cues led to the place cells and head direction cells. So this is work by mainly by my former student, Tim Liu, that did a lot of the work on the modeling and took that model, but then said, what if we compare it with a model that has a stack of images? They're still static, but they're in order. Or we have a recurrent neural net where the latent variables are part of a recurrent neural net over so many steps in time. And redid the experiment, and with Liz and Doug's input, redid the environment to be more suitable for when they do the animal and human experiments, as well as we added an attention block to the model. So then we could actually look at what features were being attended to. And we got pretty good reconstruction. So from the FPP, you'll see that a lot is the first person perspective, and GMP is the global map perspective. So in that direction, here's the inputs. There's the target image, and there's the reconstruction. So it really does a nice job reconstructing. You go the other direction, here's the input image, and there's the target image, and it's doing a good job reconstructing what it should see. So it's doing this perspective back and forth. And then when we looked at the spatial representations, we did see place cells. We did see, let me see. Okay, we did see head direction cells as before, but we also saw corner cells, border cells, boundary vector cells, object vector cells. And we did the metrics that you would have to do to make sure that these actually fit the criteria that a neurophysiologist would. One thing that was interesting, we looked for it with the grid score. We never found any grid cells that passed the level of the threshold level. So for at least this problem, grid cells were not necessary. And we actually measured things that you would measure if you're measuring place cells and things like that, and head direction cells like spatial information. The recurrent neural net analysis might be really small for you to see, but the RNN recurrent neural net version had much better spatial information, much better border cells, and this Rayleigh score is used for the head direction cells, much better head direction cells than a static model. So the recurrence really made a difference. The sequential processing made a difference in actually better neural correlates of spatial information. And then we thought, well, one other thing we didn't do before was perturb the environment. So we could rotate the arena. We could rotate the background. Or if you noticed in that video, there were these cylinders. So there were 3 cylinders. So we could rotate the cylinders, keeping everything else in place. And the arena and cylinder would be proximal cues, and the background would be a more distal cue. And we found out that the proximal cues had the most effect. So if we were trying to use the latent variables to actually decode position, if you, and I can barely see it, but if you perturb the cues that were proximal, you actually got worse decoding of position, and the reconstruction loss was higher when you did that. Now nicely, because it's a model, and we had the attention block, we could actually look at the latent variables, and these are just some attention maps, see which features these latent variables were responding to, and then ablate those particular things, like you would ablate very specific cells. And if we did that, then you really see the effect that if you ablate cells that are queuing off the wall of the cylinder, then the ability to decode position, the error goes way up, and the reconstruction loss also goes way up too. So it really shows that the proximal cues are what's important here. Now, this is part of a larger project, and I just want to give you a quick heads up on where we're going with this. So obviously our group at UC Irvine's doing a lot of the modeling work, and Liz Krastel's group at our biology department is doing work in virtual reality and imaging for this, and setting up some problems for this. And then Doug Nitz, just up the road in the Cogsci department, is looking at this in the rodent. So some early data to kind of tease what we'll probably have next year. There's work, at least behaviorally, and we'll probably do imaging soon with a human perspective taking task. So, and I hope I get this right, but people go around this town in this virtual environment, and they're looking for different stores. You can do this on a desktop or a VRK, but on a desktop, you're looking for different stores and buildings, and then you're given a test, either egocentric, which is our first-person perspective, or allocentric, which is our global map perspective. You look at a hardware store, so you're facing it, and you're told to go to the comic store. And then you have to, and you're in sort of a featureless environment, and you have to rotate so much and point where you think the comic store would be. And you can do that allocentrically, where you see this sign saying you were looking directly at the hardware store. Now, where would you point to go to the comic store? So same task, but 2 different perspectives. And what they found was not differences in the pointing error, but they found differences in when you switch, especially when you're switching from the ego to the allocentric. It took longer to actually do this task, that you had to actually do something different. So maybe a cue or a clue that there's two different processing streams for the different directions of perspective transformation. And then this is hot off the press work. Doug Nitz's group with Emily Yu in his lab have done this really clever experiment where the rat is up on a pedestal. They shine a laser on one of these wells, and then it has to go down a steep ramp. And this is what the rat would see as it's going down the ramp. And so it has to do this perspective difference. So the light is off, so it has to remember where this was and kind of transform from looking down to actually, when it gets down to the flat area, to where to go. So we think this is also a nice way of doing the rodent perspective. And if you look, they set it up so if you go to the east or west on this picture, or left or right, it's symmetric. So you're going to the same place. And they've now just done some recordings. They're working their way through the brain. This recording is in the secondary area of visual cortex of the rat, and they're getting nice cells, some showing like place specificity. So this is also very early, but actually coming soon to test some of the things that we're doing. Okay. So just a quick wrap up of what I've said. So these recurrent predictions over time actually improve performance in both the performance and also the types of neural, comparable to neural correlates that we see in the brain. The latent variables show a lot of the things that you see in different areas of the brain that are supporting spatial cognition in this very different task than a lot of people usually do. And it's interesting that some of them, I will say, some of them are not as good as what you'd see, especially the place cells in the hippocampus. So it is giving us a clue that maybe the data is pointing to other areas. We think maybe subiculum or retrosplenial, but we need to do some of the imaging and actually neurophysiology to find out if that's really true. And not surprising, but I think important to point out was the proximal cues had the most significant effect on this transformation, this task. And as we're a little ahead with the modeling because the experiments take a lot of time, but the model will be updated and extended as we get more of this data. And then last slide, then I'll take any questions you have. I have to go through this really quick. So those are our posters. So unfortunately, Tim be here. I'm really presenting Tim's work, but he'll be at SFN on Tuesday. If you want to get more details about the model or anything else, he'll have a poster session on Tuesday. And Mike is doing a poster session this afternoon, I believe, or maybe Liz is standing in for Mike. She's here. And then also on Sunday. So he did the behavioral work that I show with the hardware store and coffee shop. And he'll be presenting on Sunday. And then Harrison is doing related work. He'll be here later today to present his poster. And then he'll be presenting his poster at SFN on Wednesday. So with that, I'll stop and take any questions you have. Thank you.
So we have a lot of time for questions.
No.
As far as I understand, your error function is only for the changes of the perspective, yet you try to look at the network which changes in time. Do you have elements in the error function which deals with the temporal changes of the perspective?
Please repeat the question.
All right, let me try and understand the question. So you're asking about the loss function, and is it sequential? Is that?
Yes. Changes in different moments in time in the loss function for training the RNN.
As I recall, no. We use the loss function. We run and we did it so many iterations. So the question is, do we use the loss function over time to actually get the reconstruction loss? And not technically. What we do is we calculate the loss function, and we've done different sequences just to see what are the the optimal sequences. But we run the model with a sequence of images and run the recurrent neural net through a sequence of images, calculate the total loss through that sequence, and that total loss is what we use for the reconstruction. Yeah. There.
I think that's good. All right, so, as an animal, right, as a biological being, I somehow I can make this transformation you described from my personal perspective to the global map. But unlike your deep learning, whatever, your how do we call it? I've never been given ground truth for my global maps. I mean, sure, I've seen Google Maps, but if I were a hunter gatherer running around Savannah, if I'm a field mouse foraging, I've never seen a global map, ever. I've never been given any ground truth. I had to construct that global map entirely from my first person perspective. Can you do that, do you think? And do you think they'll
Okay.
how do we get to that?
Yeah. So the question is, in reality, there's no ground truth for your global map. And if you're, let's forget about, current day where we have really good GPS and everything on our phone, just, in the wild, we don't have a ground truth. We're kind of constructing this in our head, this global map. And this is actually surprising to me, but and us, it's been controversial with people that we've talked about this. I believe we do this. I think that's what the cognitive map is. Now, to test it with a model, we had, because we want to actually have a controlled experiment, we give it the ground truth. I do a lot of robotics, so I mean, whenever, and we've done a lot of work with navigation, much more conventional SLAM things. So some of these measurements that we're doing, and if you look at the details of this paper and others that came out of our lab, are the benchmark things which require ground truth. So you can actually do the measurement. But I agree, yeah, that people or animals, I think, are doing this, but they aren't building a high fidelity map, but they are building a map in your head. And the fact that humans, when they're some of the tasks for human navigation are, hey, go around this, like the work that Liz and Mike did, go around this, now draw a map. And they can draw a pretty high fidelity map. So you have this in your head somehow. The fact that animals, it's harder to test this. So it's impressive that Doug's group has come up with a task. But I think we're going to show it too, because I can't imagine just looking at nature. If you live in this area, we have roof rats and they're up in the trees. So the rats are doing this too, I imagine. But it's very hard to test. Yeah.
And so I'm wondering about what you talked about proximal cues being important, but I would suspect that navigation you actually want to disregard proximal cues because these are the ones that are changing more. The chair can move, a person could move, so I would actually expect the distal cues to be more important.
Yeah, that's true. So the question is, the proximal cues were important in this task, but in over a long term, proximal cues will change. And that's actually interesting. We've seen that with our robotics work, that we do robotics work on our campus and there's students everywhere. And over time, the proximal cues of students moving around get filtered out, but the stable proximal cues do stay. So the trees that are always there do stay.
So Jeff, you can keep answering questions, but Dory, would you come up?
Okay.
Thanks.
There was one back there, yeah.
I was wondering if in the preliminary human behavioral studies, they examined whether or not there was these spatial differences in perspective between male and female participants, and then whether or not your model would perhaps incorporate that gender difference that's typically seen in spatial navigation.
Oh, good question. Yes, the question's about gender differences in spatial navigation, which has been studied a lot. I'm still here. Why don't you mute? I'll unmute.
I think that was me.
Okay. We haven't done that yet. We probably have the data, so we probably will look at it, because usually people do look at that for most spatial navigation tasks in humans. Yeah.
So there was a comment when you were talking about like the lack of grid cells The question was, you mentioned like grid cells weren't particularly necessary for this task, but I was also wondering if you, like maybe the model that you're using for capable of supporting grid cells, there were some people back there supporting grid cells in the first place.
Yeah, so the question is, we didn't see grid cells, so was the model capable of supporting grid cells? And yeah, the model was capable. The way we set it up, it's completely agnostic. So all those spatial constructs emerged just by solving the problem.
Time's up.
Yeah,
Time for the
and we just didn't see grid cells when we did it.
next
for whatever
speaker.
reason. Thank you.